Okay, so we are going to apply some genetics vocab. We've covered a little bit already, like dominant versus recessive. And so now we're going to apply some more terms. So, homozygous means that an organism has two identical alleles for a gene. So, like the purple flower color that has two big P's, two of the same version that are both dominant, then that means this is homozygous dominant. So it's, as a prefix, homo means same. And then, so that means having two identical alleles for a gene. So either both copies of the purple flower color or both copies of the white flower color. And so those would be homozygous. And so usually we would specify like homozygous dominant, they're both dominant, or homozygous recessive, they're both recessive. And so if we refer to an organism that has two identical alleles for a gene, we could also call it a homozygote. So you could say it's homozygous as like an adjective, or you could say it's a homozygote as like a noun. And so heterozygous, hetero means different. So having two different alleles for a gene. So for example, having one copy the capital P and one copy the lowercase p, so a dominant and a recessive allele. The purple flower that has the big P and the little p would be heterozygous. It has the two different alleles for the gene, even though it looks like the dominant one, it has both, so it is heterozygous. And so again, you could describe something as heterozygous, or you could say it is a heterozygote because it has one of each. And so a couple more terms. Well, let's apply these first. So if this purple flower is purple because it has a big P and a big P, it has both copies of the dominant, then it is homozygous dominant. These, again, if we were to do the Punnett square, there would be two that could come out big P, little p. So one of each copy, meaning heterozygous. There's a different one. So even though they're purple, they are heterozygous because they do have the white allele as well. It's just that the big one, the dominant one, is dominant, and so that's all that shows, but it has one of each. So it's heterozygous. And then lastly, the little p is homozygous recessive because it has both copies of the white flower color, which is recessive, and so it is a homozygous recessive flower. And so we can also give terms to what we're focusing on. So if we were focusing on what it looks like, then we would be talking about its phenotype. So the phenotype is the physical appearance, like if it looks either purple or white flower color. And you can remember it as like what shows in a photograph. The phenotype is what shows in a photograph. So if I were to take a picture of these purple ones, they would all look purple, and that's their phenotype. But you might not know what the genotype is. So the genotype is the genetic makeup, is like what alleles it has. Does it have the dominant alleles, the recessive alleles, or one of each? So if you were to be asked about the genotypes of these guys and you happen to know, then you would say, oh, it is homozygous dominant, or it is homozygous recessive, or it is heterozygous. Those would refer to the genotype. And then if you were asked about like, oh, well, what's the phenotype? Then you would say, oh, it's purple, it looks purple. So it is inconvenient, bio has a lot of terms, and this may feel like a lot of terms, but it really does make something easier and faster to describe if you have one term for that situation, like homozygous or heterozygous. Like it's faster to just say, oh, homozygous dominant than, oh, it has both copies of the dominant version of the allele, and so it has big P, big P. You could just say, oh, it's homozygous dominant. So it takes practice. You can make flashcards if you're, if you need that kind of support to remember, okay, this is this and this is this. And so, yeah, now applying it again. So an organism's phenotype, you might not always be able to tell its genotype. So like all three of these look purple, they have a purple phenotype, but just from looking at it, you don't know its genotype because it could be purple because it's homozygous dominant, or it could be purple because it's heterozygous. So you don't know for a lot of them if it's, based on the phenotype, you don't know always what the genotype is. The only ones that you can know are the one recessive case, because it can only be the case that it's homozygous recessive if that recessive trait is expressed. Like if you see the recessive trait, then it has to be homozygous recessive. There's no way it could be anything else other than little p, because that's the only way it would be a white flower clot. Anything else, if there's a dominant copy, you can't really tell just by looking at it if it's both copies dominant or only one. So say you're looking at a purple flower and you're like, well, I want to know though. I want to know, is this homozygous dominant or is it heterozygous? So because an individual with the dominant phenotype could be either one, how can we determine the genotype? Is by doing what's called a test cross. So a test cross is you mate this one purposefully with a homozygous recessive individual. So like a white flower. Because if you mate this one with a homozygous recessive, it can go one of two ways. And that tells you what the genotype is based on, does this pattern happen or does this pattern happen? And then that'll tell you what the mystery individual is after you cross it with a homozygous recessive individual. So say you do a cross and it still looks the same, then it might not be heterozygous, right? It could be homozygous dominant because, well, I'll show on the slide, I'll show visually. But yeah, if any offspring display the recessive phenotype, then that tells you that mystery individual must have had a recessive allele to give. So the mystery parent must be heterozygous. So here is an example of that cross. You see a purple flower and you're like, I wonder, is this homozygous dominant or is it heterozygous? And so if you cross it with a homozygous recessive individual, then it can only go one of two ways. So if you cross them and they all end up purple, then that means that this flower was homozygous dominant. There's only big P's to give. And so even though it can inherit the little p from the white flower, if it's inheriting the dominant allele, then that's the only thing that's going to get to be expressed, and all of them will end up purple. But if you do this cross and instead you see half are purple and half are white, then that tells you that this one must have had a little p to give. It must have had a recessive allele to be able to pass that on. So sometimes you can also apply it to humans, like brown eyes versus like blue eyes. So if somebody has brown eyes, for example, me, and my boyfriend has blue eyes. So say we have kids and they all end up having brown eyes. Brown eyes is dominant, and so that means I only have both copies dominant. I would be homozygous dominant for brown eyes. And so if all the kids had brown eyes, then that means, I'm homozygous dominant. But if we have kids and half of them have brown eyes and half of them have blue eyes, then that means I must have a recessive copy of that allele. And so that's how blue eyes would be expressed, and then have brown eyes. So hopefully that also helps make sense of these patterns. Like if you were to cross something that you don't know what the genotype is, if you crossed a homozygous recessive phenotype, then that tells you, oh, this organism had this copy or this copy to give. And so Mendel, observed all these different things. And at first he was only studying one character at a time. So just flower color, just seed color. And so he came up with that law, the law of segregation, by studying just one thing at a time, even though there were two versions. And so the F1 offspring that were produced in those crosses were called monohybrids, because they we're only looking at one character, and they were heterozygous. So they were hybrids, but only for one character. So again, either like flower color or height of the plant or whatever. You're only looking at one character. So mono as a prefix means one, and hybrid meaning there was one of each allele. And so when you do a cross like this between heterozygotes, that's called a monohybrid cross. So like not a test cross, you know? If it's a monohybrid and you're crossing with another monohybrid, that's a monohybrid cross. You're crossing two heterozygotes. And so he did have a lot of curiosity, because then he was like, what if I study two characters at a time? If I look at, for example, flower color and pea color, or pea color and pea smoothness. So he identified a second law of inheritance by doing this. And so this was called the law of independent assortment. So crossing two true breeding parents that were different in two different traits, this produced dihybrids. So they were hybrids, but of two different characters. So crossing two true breeding parents that had two different traits produces dihybrids. Di means two, so for two different things, they're hybrids. They're heterozygous for both of those traits. And so when you do a dihybrid cross, that's a cross between two different F1 dihybrids. And that can help you determine whether these two traits are passed on to the offspring together as a unit or independently. Like, say, flower color and plant height. Each one of them seems to have a dominant thing, but does that mean that the dominants always go together as a unit, or no? Like, are all the purple flowers always going to be tall? Are all, any of the dominant stuff, like, is that always going together? And so that's why he did these crosses. And he was sick. So this is a dihybrid cross. He was looking at both whether the pea was yellow or green. And so yellow is dominant and green is recessive. And if it was smooth versus wrinkled. So smooth is dominant and wrinkled is recessive. And so, yeah, he was wondering, like, okay, are the yellows always going to be smooth? Like, does the dominant thing always travel with the other dominant things? Yes or no? And so he crossed these in the P generation. Get it? And then in the F1 generation, he found they're all yellow and smooth. And he was like, okay, interesting. I guess that's kind of what I would expect. It's yellow and smooth, both of the dominant things. And so let's see what happens in the next generation. Let's cross an F1 with another F1 and see what happens. And so he was like, okay, either the dominants always go together and they're just going to be either yellow and smooth or green and wrinkled. Or maybe these two different traits and these two different characters get separated on their own. And so that is what he found. So he saw that there were some, there were like a lot of yellow smooth ones, but he also saw some yellow wrinkled ones. And he saw some green smooth ones. And then he even saw some green wrinkled. And he was like, what the heck? That's crazy. And again, he did this a bunch of times. And he counted them all. And he came up with these ratios. So what he found here was not, 3-to-1. It was actually 9-to-3 to 3-to-1. So each of these is a different, phenotype. So the phenotype, if you add up all the different phenotypes that end up looking yellow and smooth, he got 9. And he added up all the ones that were green and smooth, and there was 3. and then all the ones that were yellow and smooth, or yellow and wrinkled, and they were 3. And then for every 1 green wrinkled. So this was the ratio, 9 to 3 to 3 to 1. And again, he did this multiple times. He had no life. He was a monk. And so that ratio came out like that, 9 to 3 to 3 to 1. So using a dihybrid cross like this, he came up with the law of independent assortment. He said that each pair of alleles, so the pair of yellow or green, and then the pair of smooth or wrinkled, they separate independently of any other pair. So it's not like the dominants always go together. Each allele and each pair of alleles segregates independently of any other pair of alleles during gamete formation. And so in a way, he did get really lucky, again, based on what he was studying, the traits that he was studying, and the characters, because this law only applies to genes that are on different chromosomes and different non-homologous chromosomes, or if they were, like, if they're far apart on the same chromosome, then yeah, we don't see them get passed along together. But there are exceptions. We're going to talk at the end of this lecture about, or maybe in a little bit, about exceptions to these Mendelian genetics. So yeah, he came up with this law of independent assortment, and it does apply, but only to genes that are on different chromosomes and non-homologous chromosomes, or if they're far apart on the same chromosome. Genes that are located near each other on the same chromosome do tend to be inherited together. So, Mendel's laws of segregation and independent assortment reflect the rules of probability in a way, because there's all these different probabilities, but one chance of one organism being this does not influence the chance that another offspring is going to be the same thing. Like, just because you have 4 kids, each kid is actually going to have the 1 in 4 chance. And it doesn't matter what the previous one had. It doesn't matter what the next one had. Each chance is on its own. So you could kind of think about like tossing a coin. So if you're tossing a coin and it lands heads, there's no guarantee that it's going to land heads again if you do it again. If you do it over and over and over, then it's gonna end up like a 50-50% chance. Like each time you flip it, there's a 50% chance it will either be heads or tails, and it doesn't matter what happens after or before. So in a way, that's the case too with genetics, like it's probabilities. And so if you look at like patterns, you can see how things ended up a certain way, but there's no guarantee that a generation is gonna, or like one offspring is gonna have a certain thing. Does that make sense? So yeah, in the same way, the alleles of one gene segregate into gametes totally independently of another gene's alleles. So you can think of, again, the flipping a coin analogy. So we can apply different rules to figure out the probabilities of different things happening. So there are 2 rules that can help us. The multiplication rule. So this rule states that the probability that 2 or more independent events will occur together is the product of their individual probabilities. I always hated probabilities, to be totally honest with you. But so say that you are thinking about what are the odds that if I flip this coin twice, it's gonna land heads both times. So you're flipping it twice. So each time that you flip the coin, there's a 50-50 chance, right? 1 in 2 chance that it will end up heads the first time. And then the second time, there's also a 1 out of 2 chance. So you multiply those. So if you're thinking, okay, if I flip it twice, what are the odds that they both land heads up? Then you do 1/2 times 1/2. And then so the probability is a total of 1 out of 4 that it will end up heads both times. And so that's kind of the same case here. So the probability in an F1 monohybrid cross can be determined using the multiplication rule also. So, yeah, segregation in a heterozygous plant is kind of like flipping a coin. Each gamete, so again, when they divide, each gamete has a 1 out of 2 chance of carrying the dominant allele. And then there's a 1 out of 2 chance of carrying the recessive allele. Because there's only those two options, right? It's kind of like a coin that you can flip. One example of a lab that we do in person, or can be done in person, is if you have a tongue depressor or like a popsicle stick. So on one side, if we're considering like, okay, there's two different alleles, and so there's two different sides on this one popsicle stick. One side is going to be dominant, and then the other side is recessive. And so when you flip it, it's kind of like flipping a coin of if it's going to be recessive or dominant in a gamete. So hopefully that makes sense. Maybe you have to do the lab for it to make sense. But yeah, think about it like that. Each gamete, if there's only two possibilities of like a heterozygote having gametes, then there's only two possibilities, a dominant or a recessive. And so each is like a 50-50 chance. Okay, so taking the multiplication rule. So this is an example of segregation of alleles and fertilization as different chance events. So again, anytime that you flip a coin, it could either be heads or tails. And so the chance it'll be heads is 1 out of 2. And the chance it'll be tails is 1 out of 2. So again, if we're thinking like which allele is going to be passed on in a particular gamete, the alleles are 1 out of 2 that it'll be the dominant version if this parent is heterozygous, or this gamete might inherit the recessive allele. So each of these chances are 1 out of 2. And then if we also think about the other gametes, so like an egg cell, kind of the same thing. If this parent is heterozygous and there are two, options, so then this chance of this gamete having the dominant allele is 1 out of 2. And the chance of this gamete having the recessive allele is 1 out of 2. And then during fertilization, you're basically multiplying those odds. So for each possible fertilization outcome, the chances are 1 out of 2 times 1 out of 2. So kind of like I was saying, like what are the odds that it's going to be, if I do this twice, what are the odds it's going to be heads up both times? Then each of these probabilities has to happen. So you multiply 1/2 times 1/2, and then you get 1 out of 4 is the probability that a offspring will end up homozygous dominant. It will happen to get 2 copies of the big R. And then technically same here. So are these odds that it'll happen this way, that this one will flip up this way and that this one will flip up this way. So these odds are 1 out of 2. So maybe it's a little bit more like if you were flipping 2 different coins at the same time. So how would that happen? How will one land and how will the other one land? It's kind of the same probabilities. You're multiplying the probability of each one. Again, I hate probabilities. I don't like doing it this way. I would rather just do a Punnett square, but whatever. And so that's how you could kind of calculate like, okay, if these parents are like this, then the probability that it could end up like this is 1 out of 4. Some people work well that way. And so that was the multiplication rule. And the addition rule states that if you're trying to see, the probability that any one of two or more mutually exclusive events will happen, is calculated by adding together their individual probabilities. And so you could try to figure out, okay, what if I want to figure out, you do this cross, and I want to see how many are going to be heterozygous. Or how many times is it going to be heads on one side and tails on the other side, or vice versa. So then you do the addition rule. So if we go back, then you just, okay, what are the chances that these offspring can be heterozygous? There's going to be a copy of a dominant and a copy of recessive. There's actually two possibilities that come out that way. So this one or this one. So then you would add those together, and that would actually add to 1 out of 2, 1/2, because out of all these, if there's 2 out of 4 that could be that, then it reduces to 1/2, or half of the overall probabilities that it'll end up heterozygous. So yeah, that's the multiplication, and then the addition rule is like, okay, how will that happen? And if these two different things happen, you add them up. So you can apply the rules of probability to predict the outcome of crosses involving multiple characters. So that was just one thing, monohybrid crosses, right? But you can do it with multiple crosses as well. So a multi-character cross is equivalent to 2 or more independent monohybrid crosses occurring simultaneously. Because we know that they aren't tied together, we could just calculate these different odds. Instead of doing a whole dihybrid cross, you could calculate, okay, if the odds of this happening is that, yellow versus green, and if the odds of this, smooth versus wrinkled, what are the odds that this will come out, this combination? You just multiply those odds. So you can do that. You could even do higher crosses, more than dihybrid, if you wanted to multiply it this way. Again, I just prefer it. It's something about the physical thing. So anyway, yeah. In calculating the chances for various genotypes, each character is considered separately. So again, first, the yellow versus green, that character, and then the other character of the pod shape. And then the probabilities are multiplied. So for example, if we're trying to calculate what's the probability that a seed will end up being both yellow and round, then we can multiply, okay, the probability of it being big Y, or homozygous dominant is 1 out of 4. The probability of it being homozygous dominant for round is also 1 out of 4. So then the total probability is probably 1 out of 16 that it could be this. And then if we were like, what's the probability that it will be like this? Big Y, little y, big R, then you think about, okay, what are the odds of this happening times the odds of this happening? And so that can multiply up to 1 out of 8. And then same for even more. Yeah, so you don't even have to do a trihybrid cross. You could just, again, multiply the probabilities of this case happening times the probability of this case happening times the probability of this case happening. And then that gives you the total. And then if you wanted to think more about well, what are the odds of at least 2 of these being recessive traits, then you can add up, okay, this would be a case that meets that criteria, this would be a case, this would be a case, and then you just add them. Okay, and then so that is it for part 2.